Hi, and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson, I want to look at variation. So we know that the population of a species will always show variation. Now, because we're talking about the population of a species, we're talking about all the individuals in a given place at a given time. That therefore shows us what's known as intraspecific variation. But what is variation? So variation, we're talking about the differences in observable characteristics, and we call that our phenotypes. When we look at the phenotypes, we're talking about those physical characteristics that we can see. Now, they are based on the genetic material that an organism possesses and also how the environment that they live in actually interact. So what we have to now unpick is how do these differing things have an effect on the variation that we see. Now, it's important, as I said before, the populations of a species showing variation is called the intra-specific variation. If we were looking at inter-specific variation, we would be looking at variation between different species. Why is it that a horse and a donkey show variation? That would be interspecific variation. Whereas if we were looking at the variation within horses or the variation within donkeys, that would be intraspecific variation. Okay, so if we start with the genetic side of things, then we know that our cells are made up with the genetic material held inside the nucleus. If we're talking about prokaryotic organisms, they have their DNA, their genetic material, free floating in their nucleoid. So the genetic material is present in all organisms, and we know that genetic material is made up of millions of genes. Well, what we want to look at now is the difference between different genes. So here I've got what's classed as the gene for seed color and the gene for petal color. Now you can see from my diagram, each of these chromosomes has got a different version. So if we look at the one on the left for seed color, that gives us a green seed color, whereas the one on the right is giving us a yellow seed color. At the bottom, if we look at petal color, we've got a pink and we've got a purple. So we call these alleles. If there is two or more alternative forms of a gene, we would call that an allele. Now remember, a gene is the one that's going to be transcribed and then translated into the polypeptide. So the phenotype, the expression that we see, is from that DNA sequence which is within that gene. So if you have a different DNA sequence, an alternative form of the gene, this allele, you therefore are going to produce a different polypeptide, and that different polypeptide is going to produce a different phenotype. So this is one way in which we end up with variation within a population. Another way in which this happens is when our cells go through S phase and they are going to do DNA replication, spontaneous mutations occur. If there is a mutation, in this case a gene mutation, what we're having is a permanent change to the DNA base sequence, that genetic composition of an individual, and that in itself could cause variation. So you can see from my example that on one strand, the parent strand, we have the sequence TAGC, and obviously on the parent strand on the other side we have the complementary base pairing ATCG. Well, if there is an incorrect complementary base pairing, as you can see, adenine and cytosine, they should not go together. If there's an incorrect copy made, if there's an error in that DNA replication, that could result in a gene mutation. Now, hopefully our checkpoints will have stopped this from moving on. However, sometimes it doesn't get stopped and it continues on and the cell goes through that division and so forth. You end up with this mutation, a permanent change, actually being in one of the cells that could potentially then be passed on. So then let's take a step back then. So if we think about this and we look at meiosis, our reduction division. So if you haven't already checked out my video on transcription translation or DNA replication or meiosis, then please do to find all the different informations. But if we just look at this then, meiosis is a reduction division. So the fetus goes through 2N, we have a diploid cell, goes through mitotic division many times over, producing cells within the adult, which then need to go through a meiotic division. Those 2N cells, those diploid cells, are going to be reduced down to the haploid gametes, the egg cell and the sperm cell, because that's the only time meiosis occurs. It is a reduction division. We are taking a diploid cell and we are taking it down to that haploid cell. So we have the egg and sperm. So the way in which meiosis occurs is going to result in variation. Because when that egg and sperm come back together in that fertilization, what's going to happen is we are going to end up with a zygote, which is back to being a diploid cell, which will then go through mitotic division to give us our fetus. But what's actually happened is through the process of meiosis, during metaphase 1, the way in which those chromosomes lined up at the equator, that independent assortment, resulted potentially in the variation within the species. So independent assortment is one way in which variation can occur, and this is all linked to how that sexual reproduction has actually promoted this variation, because the gametes need to be produced for sexual reproduction. So the way in which the chromosomes line up in meiosis, specifically during metaphase 1, that is going to result in potentially this variation within the population. Now we know that crossing over occurs in prophase 1 if the genes are far enough apart and as the chromosomes intertwine. Again, if you haven't already, please do check out my meiosis video on that one. So crossing over, another way in which variation can occur. And the random fertilization, which egg cell, which sperm cell are actually coming together. Remember, as the sperm cell differentiated, it has that acrosome for digesting the egg cell's protective layer. So therefore, only the sperm cell, which is quick enough at doing the digesting and fusing, therefore producing that zygote, is going to result in that passing on of the genetic material. So it is important that you remember to check out the other topic areas, because all of them interlink when it comes to understanding the bigger picture. So they are to do with genetic variation, because they're to do with the way that the genes are passed on. Environmental causes of variation are obviously to do with the environment. Now, if you look in my pictures here, you can see I have a scar under my chin. I also have a scar on one of my eyebrows. Now there's a whole story to tell on how those occurred with regards to me trying to look under a roundabout when I was really, really young, and one where I was pushed by my elder brother into a room and I tripped and cut my eyebrow on the metal frame of a bed. Great game that I was playing, but I resulted in ending up having 2 scars because I needed to get stitches done, and the healing process was a slow process which resulted in scarring. So some people, with regards to scarring, some people actually have a genetic side of things where their scars are not as visible because they are able to heal really quickly, but for other people their scarring actually is not able to heal quickly, and therefore you get more visible scars. Well, the scar actually being there is not passed on to your offspring. So, therefore, this is an environmental cause of variation. Now, this is also linked, whether it's genetic or environmental, you have to remember it's always linked to plants as well. When we talk about variation, we're talking about it with all individuals, and we tend to look mainly at plants and animals. Plants are not able to move. So, because they're not able to move, if you were to plant a seed in two different areas of your garden and they both have different amounts of sunlight, you're going to have a difference in the way in which they grow. The environment is going to have an effect on the variation that you see. So, it's important that we remember that environment and genetics come impact it together with regards to the organism. So when we look at variation, then we basically say that it shows two patterns. It can either be discrete slash discontinuous variation, and what we are saying is it will only go into a specific discrete value. Is it blood group, for example? This is one you need to know looking on further on to your other module areas. So your blood groups and genetics come side by side. If we look at the fact that you can be one of 4 blood groups, this is to do with the alleles that you have inherited from your parents. So it's controlled by 1 or 2 genes. Now, blood group is controlled by 1 gene, and there are multiple alleles for that gene. So you can have either the allele A, or the allele B, or the allele O. There are 3 alleles that give you your blood group, but you inherit 2 of them, which means that you can either have a blood group A, a blood group B, a blood group AB, or a blood group O. Now, you will learn about this more when you get to module 6 and patterns of inheritance, but for the AS content, you just need to know that this is called discrete slash discontinuous variation. Now, normally, if we have this type of variation, if we can put things into a group, are they male? Are they female biologically? Do they have attached earlobes? Do they have detached earlobes? Can they roll their tongue? Can they not roll their tongue? What we get is a discrete group, nothing in between, and therefore we represent that in a bar chart. It can sometimes also be represented in a pie chart, but we normally would draw a bar chart. And remember, if you're drawing a bar chart, your bars should never touch, and your bars should be the same width each time. That's a very important part and skill that you should know when it comes to drawing data. Now, going back, we are talking about whether it was interspecific variation between species or intra-specific variation within species. So do make sure when you are reading any question, you have identified whether they're saying it's inter or intra, because that is important for figuring out whether it is discrete, discontinuous variation, or whether it is what's known as continuous variation. Now, continuous variation can take any value within a range. So things like height of trees, your weight, foot length, height of plant. These are all results of multiple genes interacting. So we say that this is polygenic. Poly means many, many genes. It's also normally influenced by the environment. So it may be that you have inherited genes that mean that you should grow really tall because your parents are really tall, but the diet that you have been exposed to means that you don't grow to that full capacity. So it's important to remember that the environment does normally influence when it comes to continuous data. So we usually draw continuous variation data in a bell-shaped curve. Now that gives us what's known as a normal distribution. So when we look at this, we should find that we have the same mean, mode, and median. What we should also note is that 50% of our values should be at the bottom section and 50% at the top. So there's a symmetry in our bell shape, that the majority of the values are in the middle, and that the numbers towards the end are very low. And what you should also be able to note is that a standard deviation from a normal distribution is a measure of how spread out your data is. 68% of your data should sit plus or minus one standard deviation from that mean. So we would normally show that using a error bar where we have one standard deviation above or below. Now you do need to know how to use the equation. It will be given to you in the exam to calculate standard deviation. But I have and will do a whole video on the statistical test and calculating standard deviation. So if you have liked this video, then please do click on the like button and subscribe to my channel. Also check out my revision platform, www.aiqchat.com, which should help you with your revision.